Hi, I'm Al Gifari. I will explaining about DNA binding protein or DBP. DBP is a protein that binds to DNA strongly and specifically. This DNA protein interaction is a crucial behind mechanism like genetic regulation, replication, repair, and the packaging of DNA in the cell. DBPs recognize DNA through a conserved structure known as DNA binding domain. The majority one is helix turn helix, and also there are another domain that you can see here. DBPs bind DNA through hydrogen bonds, formed when a hydrogen atom is shared between highly electronegative atoms like oxygen or nitrogen. And there are three types of hydrogen bond interaction in DBPs. The first is bidentate, where hydrogen bonds occur with different hydrogen donor and acceptor, providing higher binding affinity and specificity of DBP. Another type is bifurcated bonds, and also there are single hydrogen bond. One might inquire how DBPs achieve specificity. We could answer this by understanding the DNA base and shape readout mechanism. Base readout is influenced by the availability of certain DNA bases in DNA groove. The type of hydrogen bond to water molecule hydrophobic contact between DNA base and protein is also important for base specificity. Meanwhile, the shape reading is influenced by the DNA change conformation into aDNA or zDNA, local arrangement of bases that can bend or kink the DNA, as well as the existence of minor and major groove. All of these mechanisms suspect to make certain DBP is very specific to particular DNA. While the majority of DBPs are transcription factor protein, but some perform a catalytic function like endonuclease enzyme that can cut double-stranded DNA. By a little bit exploration and creativity, we could then engineer the DNA binding enzyme to not only recognize but also cut and also replace the DNA base that we want. That is actually the idea of gene editing technology like CRISPR-Cas9 or prime editing. However, in the future, with the development of computational methods for protein design and folding problem, we may create customizable and novel DBPs, or designing an enzyme that the function not observed in nature before. This could result in gene editing technology with greater flexibility and targets, as well as robust genetic manipulation that natural DBPs cannot perform. Finally, regardless of creativity path we choose, understanding DNA binding protein or DBP will improve gene regulating and editing technology to solve problems from curing disease to fight climate change. Thank you so much for listening.